All right, let's dive into the exciting world of neurogenesis. Neurogenesis refers to the incredible ability of our brains to regrow nerve cells, which plays a crucial role in repairing damaged circuits and structures within the brain. Back in the day, it was commonly believed that neurons in the central nervous system couldn't regenerate, leading to the assumption that any damage sustained was irreversible. However, thanks to recent discoveries, we now understand the existence of neurotrophic factors. These amazing substances hold the key to repairing, restoring, and even creating new connections in the nervous system. In this lesson, we'll explore this fascinating topic in more detail. Until recently, neuroscientists held the belief that the central nervous system, including the brain, was unable to undergo neurogenesis and regenerate. However, in the 1990s, the discovery of stem cells in certain parts of the adult brain changed this perspective. It is now widely accepted that adult neurogenesis is a natural process that takes place in a healthy brain. Let's dive into lab biomarkers for neurodegeneration. They offer an objective way to track how our patients respond to our efforts in promoting neurogenesis. When it comes to monitoring responses in neurodegenerative diseases, there are several lab biomarkers that, when used in non-traditional ways, can play a vital role. These biomarkers include the alpha-synuclein seeding amplification assay, which is a CSF biomarker, so it's not really practical for our purposes. However, several blood-borne biomarkers that we discuss in detail in another lecture are intriguing. The statin C commonly used to monitor kidney disease, neurofilament light chain or NFL, and S100B protein. Each of these biomarkers offers valuable insights into the progression of neurodegeneration and can assist in evaluating the effectiveness of treatments designed to promote neurogenesis. By keeping an eye on these biomarkers over time, healthcare professionals can gain a deeper understanding of the disease's progression and make well-informed decisions about patient care.
GDNF, a protein encoded by GDNF genes in humans, is a small yet mighty player in promoting the survival of different types of neurons, including the dopamine-producing cells that are central to the development of Parkinson's disease. Currently, methods of manipulation and enhanced expression of GDNF is a very hot topic in Parkinson's disease treatment.
Building on the clinical evidence, studies conducted on animal models of Parkinson's have shed more light on how GDNF and substances that enhance GDNF expression can shield dopaminergic neurons from harmful substances. For example, the natural flavonoid chrysin has demonstrated the ability to rebalance neurotransmitters and neurotrophic factors such as GDNF, which are disrupted in Parkinson's disease. Likewise, research has shown that the flavonoid pinostrobin can elevate GDNF levels in rates with Parkinson's, resulting in beneficial effects like decreased oxidative stress, preservation of hippocampal and nigral neurons, and enhanced motor function. These discoveries highlight the promising therapeutic possibilities of strategies aimed at boosting GDNF levels.
Male mice with elevated GDNF levels outperform the control group in motor function tests. This indicates that even a slight increase in natural GDNF levels can boost motor function without any adverse behavioral effects. Interestingly, even a modest elevation in GDNF levels significantly improved motor coordination in mice, and a twofold increase in GDNF produced no side effects.
On the other hand, some forms of physical treatment like laser and electrical stimulation applied transcranially may enhance GDN activity. For example, photobiomodulation was reported to induce changes in a monkey model of Parkinson's disease, specifically changes in tyrosine hydroxylase cells and GDNF expression in the striatum, as reported in Experimental Brain Research 2017.
Soon et al., 2024, discovered that when transcranial electrical stimulation, TS, is combined with standard therapies, there is a significant increase in serum levels of glial cell-derived neurotrophic factor and brain-derived neurotrophic factor. In a study by Lee et al., 2022, it was shown that transcranial alternating current stimulation, TACS, at beta frequency, 20 Hz, boosted GDNF production in striatal parvalbumin-positive interneurons in a mouse model of Parkinson's disease. This increase in GDNF was linked to the survival of dopaminergic neurons and a reduction in motor deficits. Furthermore, Veena-Kumari et al., 2022, reported that high-definition transcranial direct current stimulation, HD-TDCS, over the right dorsolateral prefrontal cortex in patients with bipolar disorder resulted in a notable rise in serum GDNF levels. This increase was associated with an improvement in manic symptoms. Overall, the impact of transcranial stimulation on neurotrophic factors seems to vary across studies. While some research shows an increase in factors like GDNF and BDNF, others did not observe significant changes.
Mesencephalous astrocyte-derived neurotrophic factor. In this groundbreaking study, we unveil the protective effects of endogenous mesencephalic astrocyte-derived neurotrophic factor, or MANF, for the first time. MANF shields neural stem cells from injury induced by oxygen glucose deprivation and plays a vital role in regulating neuroprogenitor cell migration. When MANF protein was administered to stem cell cultures, it didn't impact cell growth, but sparked neuronal and glial differentiation. Moreover, in subventricular zone explants, overexpressing MANF boosted cell migration. In a rat model of cortical stroke, injecting MANF led to the migration of double cortin, DCX+, cells towards the corpus callosum and infarct boundary by day 14 post-stroke. Long-term infusion of MANF into the peri-infarct zone has shown to boost the recruitment of DCX+ cells in the infarct area. This indicates a promising neuroregenerative effect of MANF, as it aids in the differentiation and migration of progenitor cells, ultimately leading to an increased recruitment of neuroblasts in the stroke cortex. CDNF has recently surfaced as a promising option for treating Parkinson's disease and other neurodegenerative disorders. What sets CDNF apart from traditional neurotrophic factors are its distinct properties and mechanisms of action, making it a fascinating choice for PD therapy. Here are some key points to note about CDNF. In summary, CDNF shows great promise in the development of disease-modifying treatments for Parkinson's disease. With its distinct properties and modes of action, along with encouraging outcomes in preclinical research and initial clinical trials, CDNF stands out as a captivating area of exploration in the realm of neurodegenerative conditions. Nonetheless, additional research is essential to completely understand its capabilities and enhance its effectiveness when used as a therapeutic option for humans.